di Vienna University, Pak ya?
Betul.
Dan saat ini merupakan postdoctoral researcher di lab-nya Profesor Adrian Goldman di University of Helsinki, Finlandia, dari 2014. Bidang keahlian beliau merupakan yaitu elusidasi struktur protein dan interaksi antar protein ya, menggunakan X-ray kristalografi dan NMR ya. Nah, nanti adik-adik yang tertarik tentang bagaimana sih cara elusidasi struktur protein bisa bertanya ke ahlinya gitu ya. Kalau saya belum pernah saya mengerjakan riset tentang elusidasi struktur. Oke, mungkin itu perkenalan singkat tentang Pak Kenny ya. Beliau ini ternyata kelahirannya hanya beda 3 hari, Pak, saya dengan Anda. Bulan dan tahunnya.
Oh, gitu.
Oke. Silakan Pak Kenny untuk mempresentasikan materinya. Boleh bahasa Indonesia, boleh bahasa Inggris, Pak, terserah.
Oke. Thank you. Bahasa Indonesia, bahasa Inggris ya. Saya
aja nggak apa-apa.
Audionya Indonesia semua.
Oh gitu. Di sini nggak ada yang bahasa Inggris ya?
Ada, kita kelasnya 2 kok.
Kelasnya kelas
oh gitu.
internasional.
Oke.
Nggak apa-apa.
Okay. Yeah. I will try to speak in English, but you can also ask question in Indonesian, or sometimes also probably I will speak in Indonesian. So let's see. Because I prepared the slide in English, so I don't know if I can, translate it on the go to Indonesian. So I will share my screen now.
Rekam nggak di Holis?
Pak?
Direkam nggak?
Iya.
Bisa direkam ya? Ini Bulan minta direkam nggak itu? Terima kasih.
Yes. Can you see my screen now? So this is my second screen. Okay, wait. Okay, yeah. Okay, first of all, thank you. And nice to meet you all. Thank you to Mas Tunjung for inviting me so I can speak here in this course. And also, okay, so my name is Kenny Fidilaseris, as I mentioned before. I'm a postdoctoral researcher in University of Helsinki from 2014. It's already quite a while. So yeah. My presentation today basically about the basic concept in protein structure and function. So I will divide it into like several parts for my talk. So this is actually my presentation is, well, I don't know if it's quite long, but let's see if we can manage to finish all. And also, so if you want, I can share also my presentation after this. So so you can have it. But since yeah, there's a lot of video here, so yeah, I think it's better than like you have my presentation, then you have the PDF itself. Okay, so before I start, so basically I will tell you about my background. So I studied in ITB, in the Department of Chemistry in ITB in 2003. This is my I spent like basically my bachelor and master degree there. And in 2008, I also like as part of my master degree program, I study also in Groningen University in the Netherlands. And then since then, I moved to Vienna University in Austria to finish my PhD from 2009 until 2014. And then from there, I moved to Finland, Helsinki, to do my postdoctoral research until now. So yeah, so Finland is quite cold most of the time. So even in summer, actually, the temperature is around 20 degrees Celsius to 25 degrees Celsius. So it's not that hot. I think probably quite comparable with Indonesia. Indonesia is around 25 to 30 degrees Celsius, I think. And also, especially during winter, it can be very cold. So it can reach to like -20 degrees Celsius. And it's freezingly cold. Even the sea, you can see the picture here, it's also frozen. And you can walk on it actually. The population as for July 2019 is only 5.5 million. You can compare to Indonesian population right now. So, and also it has more lakes and islands than Indonesia actually. So around 170,000 lakes and islands. But it's very small islands, you know, it's not that big as in Indonesia. And the capital city is in Helsinki, population around 650,000 people. Yes, and here is actually my research focus since my PhD. So I mainly focusing my research on protein structure determination and also some biochemical studies, especially from process parasite causing disease such as Trypanosoma brucei, and also Leishmania and also Plasmodium. Plasmodium is the one which causing malaria. So yeah, during my PhD, I worked on Bilbo-1 proteins. You can guess actually Bilbo, the name Bilbo is from the Lord of the Rings movie, because actually this protein can form a ring-like structure in the flagellal pocket of Trypanosoma brucei. And then during my postdoctoral research, I work mainly on membrane protein. So this is one of the example is membrane-bound pyrophosphatase, which can act as enzyme which cleave pyrophosphate into 2-phosphate, and also coupled with the transport of cation such as proton or sodiums. It depends in terms of the type of ion transport. It depends on the organisms. Some organisms able to transport proton, some organisms able to transport sodiums, and some organisms actually able to transport both of them. And also, I'm also working on other membrane proteins recently from acidocalcism protein. So acidocalcism is like electron-dense organelles in parasites and basically in quite a lot of organisms, also in all living things. So this is as a storage for polyphosphates and also maintaining the homeostasis of the cells. So here are some of my research on this acidocalcisum protein, for example, VTC complex, and also sodium phosphate importer, and so on. Okay, so this is the outline of my presentation today. So basically, I will divide it into 4 parts. The first is about the basic of protein structure. I don't know, probably you already know about this, but this is some kind of overview of it, and also like a brief, after that, it will be a brief of protein function in general. And then after that, I will talk about how to solve the protein structure. There are different methods actually, like 3, the big one, like X-ray, NMR, and also cryo-EM. But I will talk mainly on solving protein structure using X-ray crystallography. And then the last, this is some kind of application because you see, like due to current pandemic on COVID-19, so I will talk about how protein structure information can help us in the battle against COVID-19. So we will talk about this if we have time. And also, if you have questions, you can ask question right away by raising your hand, or you can put it also in the chat if I can see it. Okay, so yeah, so this is the figure of a human body, well, or basically in us, human body or any organism in general is like an advanced machines. I always consider it like very advanced machines, more advanced than what the robot we have today, even where this machine, each of them, like each component of the machines basically works together in coordination and precision, right up to the smallest components. Even the smallest like component of our body, which is cells, the smallest unit of life, is packed with such a complicated subcellular component and molecules that also works in precision and in precise function and coordination to achieve goal to sustain life itself and also its progression. So here there are like some components like macromolecular components of the cells that are very essential, such as DNA. DNA is the blueprints of all living organisms, of course, and also RNA. DNA can be transcribed into RNA, and also from RNA, it can be translated into proteins. So basically, protein is the end products which do the work like the machine itself. Besides these three macromolecules, of course, there are others also, such as lipids and sugars. However, for this talk, I will focusing on the proteins. Okay. So a protein is a polymer that's made of amino acid chains. These amino acids are arranged in a specific order based on the code embedded in the DNA sequence. Okay, and this protein chains can fold, like after it's translated from the RNA, it's folded to a typical structure to achieve its specific function. So this is one of the example of protein structure. This is myoglobin structure. Myoglobin has heme. You can see here in the middle, which act as which able to like capture oxygen. And this myoglobin in general, like the function is the oxygen storage in the cells for energy generation. So if the cells need oxygen, then it can be released from the myoglobin. And in general, amino acid structure can like consist of amine group here, and also like C alpha here, and also like carboxylic acid group here, and also the side chain or R here. And this side chain actually the one which is differentiate between one amino acid to the other amino acid. So yeah, so there are 20 standard amino acids in the cells. So here you can see such as like alanine, leucine, phenylalanine, threonine, and so on. And you can see also in terms of the naming, like for example, this is the whole name, alanine, but it can be like it's the code also can be like only A. This is for a single letter code, but also can be like ALA if you want to have like a 3-letter code. And the same also for leucine, which is L, and leu, and so on. So based on the side chain, also you can like group each amino acids into like different kind of groupings, but the simplest ones is based on hydrophobicity. So based on hydrophobicity, the amino acid can be divided into 3 groups: hydrophobic groups, hydrophilic groups, and also charged groups. Okay, so for hydrophobic groups, actually this is the side chain of hydrophobic group contain hydrocarbon that are nonpolar and poorly soluble in water. So this because of that, actually because this side chain don't like water, so they tend to cluster among themselves, then interact with water. One of example is alanine, like some of the example alanine, leucine, phenylalanine. It has like more hydrophobic side chain. And for hydrophilic groups, because, well, from the name also you can say like hydro is water, philic is love water. So these side chains contain polar groups such as like carboxyl group, amino group, guanidine, and also hydroxyl groups. And because of that groups, actually, it can form hydrogen bonds with water. So that's why it can interact with water and love water. And in the protein also, quite a lot of hydrophilic residues or hydrophilic amino acids also usually in the surface of the protein, so it can interact with water. But the hydrophilic ones usually it's buried inside of the protein. Okay, then the next one is charged amino acids. So for charged amino acids, it's all of course it have charge potentials in its side chains, either like positively charged or negatively charged. So positive and negative, it can interact with each other. Okay, so this is an example of lysines which interact with aspartic acid, for example. And there is also the other ones, which is quite special. This is cysteines. So cysteines is unique amino acids because it has terminal thiol group in each side chain, which is quite reactive. So the sulfur atom here, in two cysteines, it can react with each other in the presence of oxygen or other oxidizing agent to form a covalent bond like this, which called a disulfide bond. And this disulfide bond actually it's quite good for the protein because this is one of the mechanisms for increasing the stability of the proteins. So the protein is more stable. Okay, so and then in terms of the organization of proteins, the protein structure can be organized into like four hierarchy. So first is a primary structure, and then a secondary structure, and then the bigger one is tertiary structure, and also the last one is quaternary structure. So we will talk about this in a second. So the primary structure, it's essentially the amino acid sequence coded by the DNA, as you can see here. So yeah, so and between like one residue to the other residues, there's like a peptide bond. We will talk about that later. So this is basically like examples of the chains of proteins in the primary structure. So what is peptide bond? Peptide bonds is like the covalent bonds link between one amino acid residue to the adjacent residues. And this reaction basically it's like this is a condensation reactions which produce water. And you can see here the peptide bonds formed like from this one, carboxylate react with amine group of the adjacent amino acids. And you can see also here, this bond actually it's not a normal single bond, but it's tend to have like double bond properties. Because of what? Because it has like a tautomerization with the carbonyl group here. So you can see here, it can be like single bond, but it can be also a double bond here. And because of that, actually, the peptide bonds have like a coplanar geometry. So it's flat in terms of the geometry. And because of that, also you can see like the bond length of C and N here is shorter than a typical single bond. So which is like for this C and peptide bond is 1.3 Armstrong, but for normal single bond is about 1.5 Armstrong. But these bonds also still larger, like longer compared to this CO in the carbonyl group here. Okay, and then the next thing about the naming. So usually if you see a protein sequence like this, the most left is called like amino terminal residues or N-terminal, because it has like end like amine group really, and also the most right, it's called carboxyl terminal residues or C-terminal because it have carboxyl free. Okay, and also that's also the same also for this one. So basically, this is usually what you can write in your books or in the sequence. Protein sequence. This is basically a primary structure. So the most left here, the first one is N-terminal, and the last one is C-terminal. Okay, so that's about the primary structure, and let's talk about the next hierarchy in protein structure, which is a secondary structure. So the secondary structure occurred usually due to the local conformation of the polypeptide chain, which occur mainly as alpha helix, as you can see here, or beta sheet, as you can also see here. There are like hydrogen bond there, and also the last one, which is connecting, like one, for example, in beta helix, connecting one strand to the other is turn. Okay, so that's alpha helix and that's beta sheet, and also here in between beta strands there is a turn. Okay, so now the question is why the secondary structure only like have alpha helix, beta strand, and turn conformation. So you can see that from here. So you can see from the backbone of the protein itself. So in the backbone of the protein structure, there are three main chain dihedral angle. You can see here. So dihedral angle, basically the angle between two planes like this. So you can see here, this is there's like two planes come together, and then the angle is between this one. So this is the dihedral angle. And in the protein chain itself, they have like mainly three dihedral angle. So this one is between N and amine with C alpha is pi angle, and then C alpha to C carbonyl is psi, and C carbonyl to N amine from the next adjacent residues is omega. However, as I mentioned before, actually the omega one is quite rigid, so this doesn't make like well important for determining the structure of the protein. So basically, based on those dihedral angle, Ramachandran developed this Ramachandran principle, which stated that alpha helix, beta strands, and turn are the most likely conformation for polypeptide chain to adopt. This is because most other conformation are impossible due to a steric collision between atoms. So this is, because of that, pi and psi dihedral angle determine the conformation of a polypeptide. So this one here. Okay, so this is a typical Ramachandran plot. And if you study protein structure, you must know about this because this is very important to check if, especially if you want to solve the structure. This is very important because you need to check if a specific residue in your structure is allowed or not in terms of the Ramachandran plot. Because for example, if you have it here somewhere which is not allowed, then you need to refine that region. So here you can see that each dot show a pair of pi and psi dihedral angle that occur in the protein structure. And this is actually, you can see a lot of dots here. This is like consists of about 100,000 dots taken from high-resolution crystallographic structure in PDB. And you can see typical for beta strand, it's clustered here, and typical for right-handed alpha helix, it's clustered here, and also for left-handed alpha helix usually here. And conformation with no dots basically do not occur. So it's not possible to have amino acids with psi and pi angle around here. We will learn about this later on. Okay, so this is one like example how we can show why not all psi and pi angle is possible in the protein structure. So you can see here, this is example of alanine residues, and this is C alpha, and this is nitrogen, and this is the carbonyl, and this is the peptide bond, and basically this is a part of the residues from the previous amino acids, and this is part of the amino acids from the next, and that's why you can see here there are like 2 peptide bonds, and if you see here, so this is pi between and amines to C alpha. You can see around this is pi bond, and this is also psi. So pi and psi can rotate. So that's why it's dividing the dihedral angle. But peptide bond, as I mentioned before, it cannot rotate because they are partially double bonds. So if you see this is pi 0, so basically the conformation is like this. So the dihedral angle is determined by 4 atoms. So for example, for this one here, this one, 1, 2, 3, and 4. And for phi 0 by agreement. So basically, when this one here in orientation, in cis orientation, something like this. And then if you move here, like you can move in this pi angle, either like clockwise or counterclockwise. If it's clockwise, you can see here, this is a 90-degree pi angle, and it can move further until 180 degree. So basically, in terms of the folding, the protein sequence like try to find the optimum condition where it has the lowest energy, something like that. And then this is an example of counterclockwise rotation. So you can see here. So counterclockwise there. This is minus 90 degree. And then the next one, it can goes to minus, this is minus 135 degree. And then it can goes to the minus 83 degree. So basically, so for counterclockwise, it's negative, but for clockwise, it's positive. So you can see here, because the peptide bonds cannot rotate. So you can see something like this one here. So, but for phi and psi, it can rotate. So you can see, because it's only like single bond, right? And most possible angle of pi and psi are impossible due to the clash between atom. And you can see here, this is the atom balls. It's actually quite smaller than it should be. This is basically what it's look like in real life. And you can see here, if you move like this pi angle, you can see there are some classes, right? So for example, here, if you move like this angle, it's class, and also if you move at that angle, it's class there. So yeah, so basically in protein, they need to find the optimal orientation where there is no class. So this is one of the examples. If you have psi fixed at minus 165 degree, and you can see classes occur at all phi angle except, you can see here, except from minus 170 degree phi to minus 70 degree. You can see around this one here. And then after that, it class again, and so on, and class, and so on, and then not classing. And this is if you make phi angle fixed at minus 80 degree. So classes occur at all psi angle except this one here. So this is plus 10 to minus 55 degree for alpha helix. You can see here. If it's in beta strand, then it's from plus 90 degree to 180 degree. So you can see here also. So this is in the case where if you fix phi angle to 0, and here there is all everywhere, like even if the size is rotated, everywhere classes. So that's why it's impossible. You have a protein with psi and phi, like with phi at 0, because it will be always classes. Okay, so based on that, the secondary structure have like only like alpha helix and also beta strand and also turn. So this is alpha helical structure. So this is a right-handed alpha helix. In right-handed alpha helix, you have usually like 3.6 residues per turn. So for example, from here to here, this is around 3.6 residues. And also the length between in one turn is about 5.4 Armstrong. And because of that, actually per residues, the length of the rising is about 1.5 Armstrong. And you can see also here, and this is here is the Ramachandran plot. You can see here, it's only like, for example, for this alpha helix structure here, the phi and the residues, amino acid residues, only clustered in this region there. And also you can see also in terms of the hydrogen bonding, because the local conformation form hydrogen bond. The hydrogen bond is forming between residue number 1 to the residues number 4. Okay, and this is for beta strands. So for beta strands, if it's formed like beta seed conformations, it's usually have like flat-ish conformation, but it's also sometimes it's curved also. For example, it's something like this, and it's divided into two types of beta seed, parallel and anti-parallel. It's depend on the strand orientation. So parallel, it's orient from the same direction, but anti-parallel if the direction is anti-parallel, something like that. And you can see also here, for example, for this yellow region of beta strand here, the residues clustered like around this in Ramachandran plot. Okay, how about loops? For loops, actually, it's not quite defined in terms of the Ramachandran plot, because loop can be like quite short, but it can be also very long. And this is one example here. This is a loop in the toxin protein from scorpion. And you can see here, actually, the residues here has like psi and phi angle, both in alpha helix region and also beta strand regions. That's why, actually, because of this, if we see that the certain residue have phi and psi value either in Ramachandran or beta in alpha cluster or beta cluster, it doesn't mean it's from alpha helix or beta strands. You need to check it also yourself. And for the loop itself, and the length of the loops, it can vary. But for short loop, mainly around like 4 or 6 residue long. And from there, actually, you can, like for short loop, it can be like 2 different type of loop, which is type 1 or type 2. Type 1 usually it's left-handed, and type 2 is right-handed. And this is basically also like the difference between type 1 and type 2 is usually only in the Ramachandran plot orientation of the residues. Okay, how about the tertiary structure? So tertiary structure, actually it forms when the proteins basically fall together, you know, like alpha helix, beta strand forms, and then after that it's forming like a more globular structure, bigger structure. And in this formation, basically when it's fault, it's like remove water and keep the hydrophobic residues inside, but hydrophilic residue or charged residue usually outside. Okay. However, in membrane proteins, usually the hydrophobic residues also like outside because it needs to interact with membrane, which usually also hydrophobic. Okay. So, but for the other cases, for example, this one here, like Tata binding proteins, the interaction region, it should be like, you know, like charge, because for this Tata binding protein, it's need to be charged, positively charged, because it needs to bind to the DNA, which is negatively charged. So from here, you can see that each protein has unique properties because it has like unique function also. Okay, so the bigger than secondary structure, well, it's kind of also more tertiary structure, but the smallest one, it's called like protein motifs. So this is protein motifs can be also like a part of the bigger protein structure. So this is some example of protein motif. This is like helix, turn helix. And this is also the other one here, helix loop, helix turn because it's very small loop, but loop one because it have like a longer amino acids in the loop region. And this is one of the example of the helix turn helix motifs. This is usually in the DNA binding DNA binding protein. Usually it has this kind of motifs. And for helix loop helix, example is the calcium binding domain, EF hence. So the loop actually is responsible for binding to the calcium. Yeah. And also that the other motif, this is for example is a Greek key motif, because why it's called Greek, because it's well have a repeating unit similar pattern in the ancient Greek, which we called like Greek key, something like this. So it starts from N-terminal here, going up, beta strand, down, up, and also down again. And the other one also beta, alpha, beta motif, something like this. So basically this is consists of a pair of parallel beta strands with alpha helix in the middle. So, and this motif usually found in quite a lot of protein structure that has parallel beta sheets. Also, one example is triose phosphate isomerase, which the entirely actually it's built on this motif, like several motifs, and joined together to form triose phosphate isomerase. Okay, so then we go to, well, this is like from the motif, we can see like the bigger one than motif structure. Actually, it's domains. So this is the domain is basically an independently foldable unit, and usually have like this kind of typical functional unit. And usually in domains, if your protein have like several domains, the active site can be like between domains. Okay, and usually between one domain to the other, it's usually separated by a linker region. And the other one also, this is some example of protein domains. This is mammalian proteins, which often called like bed on a string, because this is have like repeated domains like this. And with this one, basically, it's quite similar protein structure. And it has like in cells, usually it has different functions and also structure variation. It's depend on location and also in which organisms. Okay, and then the next things, the bigger one is a fold family of the proteins. So motif, domains, and then now fold family, because usually like typical most protein fold into already known structure, even when there is no sequence homologs between one protein to the other. Sometimes they have quite similar homologous structure. From there, actually, we can conclude that structure preserves much longer than sequence. And there are three common folds, usually with beta alpha barrel, 7TM receptor, and also 4 helix binder. But of course, also there are other like unique fold. For example, this one here, this is membrane-bound pyrophosphatase, which is only consists of alpha helical structure, like 16 alpha helical structure bundled into one structure. And this is quite unique. Okay, so this is for beta alpha barrel. You can see here. So for beta alpha barrel, it has like beta strands and then going up helix and then strands, helix and then strands again. So it's have like beta alpha beta motif, as I mentioned before. And these folds usually are unique to about 40% of proteins, and mostly actually usually it's enzyme. For example, even like TIM barrel, this one, or even like for alpha amylase also have these properties. And also the next one, the common one is 7TM fold. So for 7TM folds, example is bovine rhodopsin. And not only this, actually this kind of folding occur in more than 500 proteins in humans. And it has like, this is a membrane protein, it has 7 transmembrane, alpha helical transmembrane here. And mostly actually it's act as a signal transduction. Okay. So that's about the foldings and about the tertiary structure. And how about the quaternary structure? So the quaternary structure, basically, if you have different separated proteins and interact together to form one bigger structure, and to have like a certain function. This is one of the example of hemoglobin. So basically, for tertiary structure of hemoglobins like formed together, and then it can like capture oxygen from the lung and then transport it to the cells by bloodstream. Okay, so that's the quaternary structure. And how about the protein representation? So for protein representation, so the protein structure, it can be like represented quite differently. It depends on what you want to do with it. This is some of them actually, but not limited to this one in terms of the protein structure representation. This is ball and stick. So basically, it's each atom is the ball representation, and then the bonds is stick. You can see something like this. It's usually very crowded. Or if you want to have it simpler, then you can only have C alpha trace, something like this. You can see also here, like the trace of alpha helix, you can really see here, and also loop, where is it? Or you can also represent it, the protein structure only with stick, okay, something like this. But the most common, actually, we usually represent the protein structure in cartoon, because from here you can see like the secondary structure, for example, alpha helix and also beta strand, and also turn here, and it's easier to see. Or if you want to show like surface area also, surface region where interact with water and so on, so you can see depict your protein in this surface representation also. Okay, so this is basically about the basic of protein structure. If you have any questions up to now, you can ask now.
Okay, thank you, Dr. Kenny, for your great and good presentations. I hope all students will be participate actively in this guest lecture, because Dr. Kenny already explained very good, I think. Is there any question from the students? Okay, maybe I will ask.
Yes.
Dr. Kenny, when I was studying in Yamaguchi University in Japan, my supervisor asked me to study about protein interaction, but at that time I don't know how about the research step. Could you please explain about the research step to study protein interaction?
Yes.
Maybe
so,
this is your topic.
Yeah. Well, we can discuss more about this. So, but basically, there are like for protein interaction studies, there are different kind of method. So, but of course, for that you need first you need to express the protein separately and purify it. I mean, this is the most common if you want to do it biochemically in a pure protein. And there are different methods to do that. For example, ITC, isothermal calorimetric method. So where basically you have one protein in the syringe and the other one in the below, in the kind of what is this like? So basically it's below. So basically you inject your protein from the syringe down to the compartment where the second protein is located, and from there actually you can see like that you can like monitor the heat release because of the protein interaction, and then from there you can calculate also the KD, the dissociation constant, and also enthalpy and also entropy of the interaction and so on. And even you can also see like how much energy in total for that protein interaction. That is one example. There are other examples, even in NMR. For using NMR also you can study that. But the simple ones also you can do like just, well, either pull down assay where you can monitor also with SDS page. So that's also like the simplest one. And so basically there are different kind of method. We can discuss about this later on if you interest more.
Okay, maybe if I have a project related to this research, maybe I will contact you.
Yes, please.
Actually,
Yeah,
I don't know nothing about the protein structure or knowledge, so it's so sometimes it's a challenge for me to do this kind of research. Thank you, Dr.
Yes, my pleasure. So yeah, I mean, if you have any questions or anything related to protein structure in general, or protein interaction, or in general like more biochemical studies, you can also contact me, like even after this lecture. And also for students, it's the same. If you have any question regarding my presentation or structure in general, you can also contact me. I think, yeah, I can give you my email address also. Okay, so yes, if there is no more questions, I will go now to protein function. This is just brief about what kind of protein function in general. So as I mentioned before, that 3D structure usually 3D structure of the protein usually determines its structure, right? So there are different kind of function based on the 3D structure. This is one of the examples as a defense mechanism for antibody. You can see here, like antibody usually it has like typical Y-shaped structure, and it's they are like usually receptor binding domain which binds to pathogen. In this case, for example, a virus. So something like that. And also the other ones, it can be also for communication. This is one. The example is insulin. It's quite a short protein structure, which like express when there are a lot of glucose in the bloodstreams, and the insulin expression basically trigger the cascade mechanisms to basically tell DNA to express a glucose transporter. Therefore, actually, it's expressed in the membrane there, and then it will like import glucose from the bloodstream inside to the cells. And the other one also for as an enzymes, a lot of proteins actually act as enzymes. This is one example, is alpha amylase. So alpha amylase basically the enzyme that cleave amylose into glucose or short chain sugars, as you can see, something like this. And actually, it's also abundant on your saliva. That's why actually, if you like eat rice and chew it quite a long time, and you can feel like sweet at the end because of that. And the function usually also for transport. This is one example of protein transporter. This is a calcium transporter, calcium pump. So for this pumping, actually, the calcium pump needs magnesium and also ATP, because it needs energy, and the energy is from ATP. So basically, it needs magnesium and ATP to move calcium ions back to sarcoplasmic reticulum after each muscle contraction. So this is usually in the muscles. And the next one also for storage. This is an example of a ferritin. Ferritin is a spherical protein with channel that allow the iron atom to enter via specific channel here. And this is quite abundant in different organisms. And so inside ferritin, basically, you can see it's quite hollow structure. You can see there, and the iron which is moved inside the hollow region is like oxidized, because outside is usually Fa2+, which is usually quite toxic. So if it's stored in ferritins, it's oxidized into Fa3+, which is non-toxic. So this is also one mechanism to sort iron, and also, you know, to avoid damage because of iron toxicity in the cells. The next one is about structure. This is example of collagen triple helix. So collagen form a strong triple helix that is used in the body for structural support. So you can see here, like these triple helix collagens can like forms more oligomer with each other, aggregated to form collagen fiber. And this type of collagen, actually, you can find it in skin, quite a lot of it, and also in tendon. So that's basically more or less brief about protein structure. And there are many other protein structure out there. Okay, and now I want to tell you about how to determine the protein structure. So there are mainly, like the most common, there are 3 different methods for determining protein structure. So either via X-ray crystallography or NMR or cryo-EM. But we will focus during this talk is only determining protein structure with X-ray diffraction. So for X-ray diffraction, so you need to have crystals and then put the crystals under like X-ray beam, and this crystal will diffracting the X-ray beams, and then basically you collect the diffraction pattern in the detector. So we will talk about this more. So yes, this is the phenomena. So first, the need is you need to have a protein structure, proteins crystals, put it into the beams, and then it will diffract it, and you collect the diffraction. So to get how we produce protein crystal, actually, of course, this is the workflow, like the common workflow to do X-ray diffraction experiments. First, we need to clone our gene of interest into specific expression system, and then express the protein and do purification. So this one here, this is the first step. So for this one, of course, nowadays it's quite easy to express protein recommendedly in a host cells, and but each proteins sometimes have their own difficulties. Sometimes they have difficulties in the expression stage, sometimes also like cloning stage, and so on. And also, for example, if your protein have lower expression in E. coli, for example, when you try to express it in E. coli, the yield is very low, then you need to choose probably other expression system, for example, yeast or insect cells, or even mammalian cells. Once you can express your proteins in high yield, then you can try to do protein purification. There are different kind of method for protein purification, and yeah, I will not talk about this this time, but basically you need to purify the protein, and then once you see your protein is pure, then you can try to characterize it. For example, if you have a enzyme, so to see if your protein is good enough, folded well and active, you need to do some activity assay. So yeah, so that's the next thing. After you purify your protein, you need to check the activity, because if it's not active, then something wrong. Probably the folding is not quite good, or aggregated and so on. So you need to go back again and try to express it again, for example, in different hosts and so on, or you try to optimize the expression system and so on. So if not successful, and then go back again. But if once you get pure protein after purification, then you can start doing a crystallization trial. So for crystallization trials nowadays, we use robots. So basically, this is the plate where you put your protein here. You see this tiny hole here. This is 2 each, and this is like the big well here. So you put basically solution, crystallization solution here in the well, and then you put your protein, pure protein here, and also here. So basically, usually you put like a protein solution together with the well solution, like in 1-to-1 ratio, for example. And we do that usually not manually, but using robot. This is show you the robot in action. So in the needle you have a protein, and in the well you have a crystallization solution. So basically this is how the robot like put each protein drops into the well, and then from there you basically incubate it at a certain temperature, either like room temperature or lower temperature, for example 4-degree room, it's depend. And then you need to inspect it like regularly, but this time actually you have a robot also to do that, but you can do also manually, for example here under the microscope, and then you can see, you know, like each day how the crystallization growth happened. So you can see for in this video, for example, it's growing like over, well, the protein grows, the crystal growth can be like 1 day, weeks, or even, you know, months or years. It's depend on the proteins also. Okay, so basically this is why you need to form to grow crystal, because in the crystal, so basically your proteins arrange in certain way, you know, like a periodic way. So that's why actually when you put it into in the X-ray beams, then it will diffract. So that's the idea. So once you got crystals, then the next thing you do, you need to do data collection. And for data collections, we can do it at home source in the lab if you have like home source. This is, you can see here in the left, basically this is typical like home source X-ray generator here, and basically you put your sample. This is in the plate here, and this is the beam, X-ray beam from here, and it will, you know, put it into your crystals. And you can see here, basically this is usually like the nitrogen stream to make the crystal cold usually, and the detector actually you can find it here, this one here. But usually for home source X-ray machines, the energy is quite low, so it's not that strong. Sometimes, you know, you can get a nice result also here. You can get a protein structure to high resolution, but sometimes not. So for that, usually you need to have like a stronger X-ray beam, and for that actually we go to synchrotron. So this is one example of synchrotron in United Kingdom. This is a diamond light source. We also here usually send crystal there. So this is the synchrotron where you accelerate the electron here in this big building here. So the principle is that basically you generate or you produce the electron and then accelerate it here in this smaller ring. Once it's reach like almost the same as speed of light, and then it will move to the storage ring here, where it's like the electron will, you know, like circling around, and in doing so actually it will emit it X-ray beam, and then so basically we collect this X-ray beam and then use it for X-ray diffraction of your protein crystal. So this is the video of it. Okay, so you see like accelerated and then storage, and then you can see here the beam projected to each like a hatch for protein diffraction. Okay, so that's the how X-ray beam generated in synchrotron, and this is basically the system how we put your proteins crystal into the beam. So basically this is the sample exchanger, and then it's used robot to pick the samples here into the X-ray goniometer head, basically this one here, and you can see there. Wait a minute. Yeah. So you you can see here. This is actually the loop, and this is actually your protein crystal is here. It's very tiny, and the dimensions usually only around like 50 micrometer. It can be up to 300 micrometer, even sometimes 500 micrometer. But to get your protein crystal into this loop, basically you need to visit yourself. There is no automatic way to do it at the moment. So you need to try to visit yourself under a microscope. So you can see here, see, and this one here, you can see this is the nitrogen stream, because you want to have it cold during measurement. Why? Because when you radiate the crystal with X-ray beam, it's basically will destroy your protein crystal, especially if you do it at room temperature. That's why if you put it at cold, so the damage will be minimized, at least for a certain time. Okay. And here, basically, once you put your crystal in the loop into the goniometer head, and then you can check also, in the monitor room here. So you can see if your crystal is indeed in the loop or not. You can see here, actually, you can see the crystal there. And then from there, if you have everything, your crystal there, and then you can start shooting your protein crystal with X-ray beam. So you shoot with X-ray beam, and then you collect the diffraction pattern in the detector. So this is one example of the pattern, X-ray diffraction pattern. You can see a lot of dots. These dots corresponds to the protein in the crystals. So basically, when we collect X-ray data from the protein crystal, it's not only like this one image, but we collect a lot of images. So it can be like thousands images. So what you can see actually during collection is something like this. So this is basically like a movie of images collected in X-ray data collection. You can see here. So once you got the data set, so what you get actually from the dot there, you can build the electron density. Okay, so so data collection, and then you build electron density from the data you got, and then from there basically you fit into electron density your protein structure. So because you know the protein sequence, right? And then you try to put it one by one, like each residue into the electron density. Okay, so so you see, you try to fit each residue into electron density. And so this is basically, it's called like a model building. And then after that, you can have like model structure of your protein. And then from there, you can analyze further for example, for protein-protein interaction, or if you want to do mutation and so on, you can do more research from the result. So that's basically how we solve the protein structure using X-ray diffraction in brief. Okay. Is there any questions? No?
Is there any question? Maybe they're still thinking.
I have a question.
Yes.
Kunjung, please.
Okay, so Kenny, thank you for the wonderful presentation. I think you have more before you continue, I think I'm interested in knowing whether there are differences in the common folds for membrane proteins versus non-membrane proteins. Are there some folds that are adopted exclusively by membrane proteins and not non-membrane proteins? And what are the tendencies of proteins forming crystals? Do you have any, I guess you know from many crystal data, some folds do not, it's hard to get crystals. Yeah, thank you.
Yes. So yeah, what is the difference between in terms of the structure or the fold between the membrane protein and with globular protein, right? So the most difference basically, usually for membrane protein, it has usually like alpha helical spanning bundle, like alpha helical bundle, as I mentioned before, for example, rhodopsins. So basically, it's always like alpha helix, but sometimes also it's barrel structure, like for example for beta barrel structure. So it's just like barrel but with beta strands up and down. And one common feature for membrane protein, it's always have like hydrophobic residues in this outside where it's facing the membrane. That's basically it's that's the most common. Usually for membrane protein, either alpha helical or beta strand, but surrounding it, like outside in the membrane facing region, you have hydrophobic residues. That's mostly. But for soluble protein, that's not the case, because if that happens, if you have hydrophobic residues, like a lot of hydrophobic residues outside in the surface, then it will not soluble. It will aggregate it or forming, like interaction with each other. So that's the thing. Usually for globular protein, you have mostly like hydrophilic residues or charged residues in the surface. And so for the globular proteins, it can have like many different folding, many different conformations. It's quite a lot. So yeah, it can be anything. So for the second question is, sorry, I forgot.
So the tendencies in forming crystals. I often hear that membrane proteins are difficult to crystallize. Is it because of that particular fold? I mean, the fold is limited to helices and barrels, and with hydrophobic
region, yes,
jacket or something like that.
Yes, correct. Well, actually not because of that. For membrane proteins, for the folding, usually not really determining if your proteins is able to crystallize or not. The problem with membrane protein, actually, because when you extract your membrane protein from the cells, from the membrane cells, okay, so basically you need to remove the membrane, right, surrounding membrane. In doing so, actually, you need to add detergent. So detergent will interact with the hydrophobic region surface, which previously interact with the membrane bilayer. And usually this detergent is not quite, like well behaved or ordered well. Okay. So that's why actually sometimes it's quite difficult, even if you can crystallize your membrane protein. Usually in general, the diffraction resolution is very poor compared to soluble protein. For example, if soluble protein, you have a really nice resolution around 1.5 Armstrong or 2 Armstrong. For membrane proteins, probably you can just go to like 3 Armstrong. So the higher Armstrong means poor resolution. So it's not as good in terms of the structure resolution compared to 1 Armstrong or 2 Armstrong resolution. So that's also. And the other thing, for in general, if you have like flappy region, either like long loop, which is not like stacked together, not rigid, but it's flappy, then it will be quite difficult to crystallize because it will hinder the contact formation between one protein to the other. So that's why actually when you want to do crystallization, if your protein has his tag, for example, his tag is quite waving around, right, in the solutions. So you need to remove that. You need to remove that first, and then you see your protein is rigid enough, then you can start to do crystallization. And the other thing also, there's other method to do if you have, for example, a certain region of your protein is quite flexible. For example, this is your protein, but this region here in the middle here is very flexible. So what you can do, you can cut it with trypsin, for example. But so basically you do like trypsin serial dilution. So you have your proteins and then add like trypsin in different concentration, and then just set up crystallization. So somehow trypsin managed to cut only that region and then keep the rigid proteins intact. And from there, you build protein structure.
So the protein doesn't need to be pure. I mean, just one protein. So you can add, you can treat it with other protease, and then you put it in the.
Yes, because the protease is a small quantity in terms of the quantity. So to make for crystallization, the purity is important. Usually, if you have like more than 90% purity, it's really good already. Okay, so even if you have some small, impurity, it's still okay. It's no problem. Actually, I have a case during my PhD. I have a protein. It's not pure enough. Actually, I have like two bands, like equally, like the top and the lower band, equally. And I just set up crystallization because I didn't have anything to do anymore with these samples. Okay, and I got crystal only from one of them, like the lower band, which is crystallized. So I mean, crystallization is a trial and error, I think. So sometimes, you cannot really say if your protein will be crystallized or not. It's depends. Okay. And also in terms of the concentration, like the general rule is that you have at least at the beginning when you start a crystallization experiment, you have like, for example, 10 mg per mil. That's the general rule, but it doesn't have to. Sometimes you need even like 30 mg per mil of protein, pure protein. Sometimes you just need 1 mg per mil. So that's why you need to always optimize your protein concentration and also your crystallization solution and so on. So there are a lot of different things to consider.
You need to be lucky, I guess.
Yeah, that's the most important thing, actually.
Biology is a mess.
I think all biological science experiment, Pak Tunjung. Lucky is those.
Yes. Thank you. Yeah, you can continue. Thank you.
Okay, thank you. Is there any other questions?
For students, please, if you have any question regarding to the topics like a protein structure and function, or also related to X-ray crystallography, you can ask directly to Dr. Kenny.
Yes. So if you don't have question now, you can have it later. Just email me or contact me. Yes, you can find me. I mean, well, in Google actually, if you find my name, I think it's only will be directing to me because I am the only one in the world who has that name.
Oh, good.
So yeah. Okay. So next we will talk about, well, this is the rest. So once you solve the protein structure, and then you need to like submit it somewhere, right? So this is the bank for collecting all the protein structure solved either by X-ray or by NMR or by cryo-EM. It's called like a protein data bank. And up to last year, So this is about 160,000 structure deposited there. That's quite a lot, actually. And in terms of the method for solving structure, so 88% is by X-ray diffraction, 8% is by NMR, and almost 4% by EM. But actually now, cryo-EM is very, very promising method for solving structure. A lot of protein structure nowadays, especially the big one, like above 100 kilodalton, solved by cryo-EM. And also in terms of the resolution, they can go to highest resolution, same as X-ray. For example, like 2 Armstrong. 2 Armstrong for X-ray is really good already. And for cryo-EM, like long time ago, like some years ago, not really long time ago, some years ago, it's really difficult even to reach like 4 or 5 Armstrong resolution. But nowadays, with cryo-EM, you can reach like even like 2 Armstrong, even lower than that. So yeah, so you can see also here, in terms of the amount of structure deposited by cryo-EM, it's exponentially growth. So yes, so actually cryo-EM actually quite something in the future for future. But of course, even cryo-EM also good, but some protein cannot be solved by cryo-EM, but can be solved with X-ray, for example, or NMR. It depends, it depends on your protein. For example, in my case, I had like during my PhD, I tried to crystallize one of my proteins. It's not successful until after like 1 year, and then I tried it with NMR, and then it's working fine. So yeah, it's depend on your protein. Okay, so next, actually, this is the last part of my talk. This is about how protein structure information can help us in the battle against COVID-19. So basically, this talk more or less about the application after we have protein structure, what we can do next, something like that. Of course, it's not only for disease. I mean, for agriculture also can be used. For example, this membrane-bound pyrophosphatase, the one we working on here. This protein actually can be used in agriculture to improve agriculture, the crop yield, because this protein actually, like expression of this protein makes plants more stress tolerant. For example, during drought. So this is quite good. So that's why actually in some lab in Japan, I think they try to try to use this protein for agriculture. So this is one example in the battles against COVID-19. I choose this because this is quite hot topic nowadays. So I think it's quite interesting. So yes. So this is, well, I think you already know the background already, like how COVID-19 already originated from Wuhan, and then basically what virus causing this disease, which is SARS-CoV-2, and what symptoms you have. And also the other thing, you can see also the similarity between SARS-CoV-2 with other like previous pandemic causing SARS-CoV, for example, like MERS or also SARS. So with SARS-CoV, like this is SARS-CoV is, I think, the 2002 pandemic, or the similarity in terms of the genome is around 80%. With MERS is about 50% and so on. And it has the most striking, like high similarity with bat, actually, is SARS-CoV-2. It's about 96%. And with pangolin coronavirus, also about 85 to 92%. That's why actually some researcher or scientists consider pangolin as a reservoir of SARS-CoV-2 between like bat SARS coronavirus and human coronavirus. So reservoir before it's jumped to human. Okay, so Okay, so basically, this is how SARS-CoV-2 replicates in its host. So it's infect a host and then interact with the receptor in the human cells, in this case, ACE2, and then from there, it's released its genes, and then it's used like human machinery, cell machinery to amplify itself. And then after that, it's released from the infected cell and then goes to the next one and so on, right? And so basically structural biology, or if we know the protein structure of this, for example, the spike proteins of the SARS-CoV-2, we able probably to understand the mechanism how the virus infect host cells. And also the next things, we can also probably develop drugs if we know the proteins are responsible for like, for example, replicating its DNA and so on, its RNA and so on. We can develop drugs against COVID-19, or even because, you know, once the virus like enter our body, so basically our body will produce like immune system, right? And this immune system basically will generate antibody and so on. And from here also we can try to understand how antibody we produce inhibit viral infection and so on. So we can use this also as potential drug, or, you know, or in developing also for development of vaccine also, that's also possible. So yes, so basically this is the idea. Information of protein structure is needed to understand how the virus works and how to fight back. Okay. Okay, now we will talk about how does the virus infect host cells from the structural perspective, of course. So we know that in order to enter host cells, the virus need to interact with here. You see this one here. This is ACE2 receptor. Should be interact with the spike protein of the virus. So this is actually, it's like, you know, opening the door of the cells. So ACE2 is the door holder, and S protein is basically the hand of the virus to unlock the door. So ACE2 is an enzyme attached to the cells of membrane located in lungs, arteries, heart, kidney, intestines, and quite a lot, even in basically, well, everywhere in your brain also it contains that. The function of ACE2 actually to lower blood pressure by catalyzing the hydrolysis of angiotensin 2 into angiotensin. So yes, so since the ACE2 itself basically everywhere, so that's why actually the symptoms of COVID-19 can vary between person to persons, you know. So when the first time it's came out, like the virus, it become outbreak, actually no one knows about the spike protein. But then like some months later, actually like the first structure of spike protein came out. So this is the structure of spike protein. This is attached to the surface of the virus. And this is the side view, and this is the top view. And from there, you can see that the proteins have like trimeric conformations. And you see here, like this is the N-terminal domain. It starts from here, and this one here. So this is basically the S1 domain, the whole, and this one here is the S2 domain. And what unique about the S protein from SARS-CoV-2 compared to the SARS-CoV, actually it has furin cleavage site. So this cleavage site actually makes the virus more virulent. Okay, and also the other one, also this RBD domains. RBD is receptor binding domain. This RBD is the one responsible to attach to ACE receptor of human cells. Okay, so that's from the side, top view, and basically this one here, the structure of partially open form of the S protein. So basically, when the S protein interact with the ACE protein, so this RBD domain should be exposed outside. And this is a structure actually where it's one RBD already exposed. So from there, actually, so you can see here how mechanisms, how it's open. See something like that. Okay, so this is the first one, but then also the second one, and then the third one. Actually, now actually we have like structure protein even where like all three RBD domain actually exposed and open and interact with antibody, but I'm not showing you here. So yes, so once it's open, so it will interact with SE2. Okay, and from here you can see like you can understand with knowing the structure what is the difference between SARS-CoV-1 as protein with SARS-CoV-2 as protein. You can see also because SARS-CoV-1. Okay, so here, well, this is basically the interaction, the residue which important for interaction between RBD of S protein and SE2 of human cells. And the KD, the dissociation constant, is very low, 4.7 nanomolar, which means that the interaction is very tight. Okay, but if you compare with the SARS-CoV-1, actually, you see, because SARS-CoV-2 is more virulent, it means like it's more strongly attached and then open and then insert to the cells, the virus, compared to SARS-CoV. That's why actually SARS-CoV, the pandemic is not as severe as SARS-CoV-2 or COVID-19. This is one of the explanation, because you can see also in terms of the dissociation constant, how tight the interaction between RBD domains with the SE2 domains. For SARS-CoV, the first one is quite low, around 31 nanomolar, compared to SARS-CoV-2, which is about 5 nanomolar. So the lower the KD, tighter the interaction. And this is with the, if we solve the structure, we will understand why the SARS-CoV-2 RBD interact better compared to SARS-CoV-1 RBD. So there are like three different the difference in terms of the interaction. There are some, like, basically what I will say here is that residues in SARS-CoV-2 make better interaction with the ACE2 compared to RBD SARS-CoV-1. Okay, so that's why it's tighter. Okay, next, how does antibody inhibit viral infection? So when we got infected with the virus, our body will fight back with producing antibody, right? But how actually this antibody inhibit viral infection? One way actually it's inhibit the interaction between S protein of SARS-CoV-2 with ACE2 receptor. Okay, so then there are a lot of research also try to understand this by solving the complex structure between S protein in complex with the antibody generated by human. So this is the structure where RBD interact with SE2, and this is the receptor binding region here. You can pay attention here, and then actually this is antibody produced in human, which also interact in the same region as SE2, but somehow slightly different. You see, and this is B3H also, CB6 FAB also interact in the same region. But sometimes actually, even it's neutralizing antibody, it's interact somewhere else, like not really in the binding region with SE2. For example, this one, reg N 10987, it's more in the site. And sometimes actually this one here, CR3022, actually interact really like somewhere else completely different. And for this one, 488, actually not interact in the receptor binding domain, but with the N-terminal domain here, you can see. So yeah, so from here you can understand why certain antibody can be neutralizing, why some others not. If we know the structure and the complex structure, then we can understand that. Okay, and this is example actually the antibody resistance of SARS-CoV-2 variants B117. This is the UK variant, and also B1351. This is the South Africa variants. I mean, I know nowadays there are a lot more variants, right? So, but this is just example how if we know the mutation happened in these variants will influence the resistance of SARS-CoV-2 to these antibodies or vaccines actually. So you can see here there are some mutation in variant UK variant. This is N mutation, become asparagine to tyrosine mutation in RBD, and N-terminal domain there are like 2 deletion. For South Africa variant, actually you can see here like in RBD domain there are like 3 mutation here, and the same, these 2 deletion in N-terminal region. And you can see here also actually antibody directing RBD. It's not quite neutralizing anymore for South Africa variants compared to UK variant. You can see here. But all MIB directed to N-terminal domain, which is this one here, all actually not like become resistant. This is, I think, because the deletion is basically like change the overall conformation of N-terminal domain. So that's why it's not, it cannot interact anymore. So this is one of the mutation in the RBD, which causing not only steric classes, but also a charge change. See here, this one. So this is the mutation from glutamic acid 484 to lysine. So from negatively charged to positively charged. Then that's why it's become resistant. And this is a mutation in the N-terminal domain. So basically, because there is some changing in conformation, that's why it cannot fit anymore, the antibody, to the N-terminal domain. Okay, so even to the vaccine, actually, this is the Pfizer vaccine, I think, and Moderna vaccine. Oh yes, it's mentioned here, Moderna vaccine and Pfizer vaccine. So South Africa variant actually become resistant to both Moderna and Pfizer vaccines, but not for UK variant. And even for convalescent plasma, it's also the same. It's more resistant to South Africa, but not to UK variant. Okay, so the next thing is how about the finding drug which able to inhibit the viral infections. There are a lot of research also on this one. So about how we can develop drugs against COVID-19. So the most important, one of the most important actually, the protein which is as a drug target is main protease. So this main protease is responsible for processing polyproteins, expressed from the RNA genome of the virus. So basically this polypeptide, before it's folded and assembled into a new viral structure, it needs to be cleaved or cut using this one, main protease. And this protein actually absent in human, so that's why, because there is no homology in human, then if we able to inhibit the action of this main protease, then probably it's only inhibit the main protease but not human protein. So that's why it's attractive drug for targeting SARS-CoV-2. And there are like inhibitor for that apparently, and it's the complex structure of inhibitor. It's called like N3 inhibitor, with the main protease is already solved, as you can see here, and the N3 inhibitor is bind basically here in the allosteric region of the protein. Yes. So okay, so N3 binds to the substrate binding pocket, and you see here it's extended to conformation. And then from there, they try also to find other compounds which able to inhibit. They did a virtual screening, and they found like this one, sinanserin, with the IC50 around 120 micromolar. And the structure is like this. There is no structure of X-ray structure about this yet. But then also they try to do high throughput screening. So basically they screened a library of about 10,000 compounds. So this is done in the lab, and check among these 10,000 compounds which ones were able to inhibit and binds to the M-pro of SARS-CoV-2. And they found like 3 others compounds. With the best one is this one, the glucib, with the IC50 around 1.5 micromolar. And you can see based on the docking, actually this one here, how the compounds binds to the active sites of the protein. And then this is a new type of inhibitor. This quite complex structure here, and they managed to solve also the complex structure of these compounds with the M-pro structure. And you can see also how it's interact. So basically from here, once you know the complex structure, once you can solve the complex structure, then we can, like extend further the compounds. For example, okay, probably you can see, for example, here, there is some region here which is not occupied yet, and also there's some kind of cavity. Then probably you can extend the compound here, like adding alkyl or anything. So then it can, like bind tighter and more potent. So yes, and also for these compounds, actually they check also the antiviral activity in veroasic cells, and you can see like the IC50 is around 0.5 micromolar, and the other one is around both around 0.5 micromolar. So yeah, so this is basically how people try to develop a new drug based on the protein we know. Okay, and this is the other ones, the last ones about how remdesivir inhibits SARS-CoV-2 infection. If you read news like some times ago, they found that this remdesivir actually able to inhibit also this RNA-dependent RNA polymerase enzyme and inhibits SARS-CoV-2 infection, right? So remdesivir is a modified adenine-like nucleotide that is converted into the active 3-phosphate form in the cells. And then when it's activated, and then it can be like added to the RNA strand here. You can see here the yellow one. But then when it's added there, then the transcription, translation process, replication process actually stop, because this is not quite compatible. So that's why it's not, it's not polymerized any longer. Okay. So this is the idea. So yes, so basically that's, and there are some interesting facts since the beginning of the pandemic, actually. So since last year, like 2020, so the end of 2019, December, we know about the pandemic. And then actually about in February, the first structure of protein structure from SARS-CoV-2 and solved. And since then, until, well, during last year, more than 10,000 protein structure already available in solve. That's how advanced the research on this actually. And also, since then, structural biologists have visualized most of the SARS-CoV-2 protein, I check also, like, yeah, indeed, quite a lot of protein, especially the structural protein, like the viral structural protein, they already solve most of them. So some of them like spike protein, as I mentioned, main protease, and then papain-like proteinase, and also which those actually are promising for drug discovery leads. Okay, so basically that's it. As this is just a summary about the role of structural biology for the fight against COVID-19. So yes, so that's my presentation. If you have any questions, we can discuss.
Okay, thank you, Dr. Kenny, for great presentations. We still have around 12 minutes to 4 p.m. So please, if you have any question for all the students here.
Yes, if you have question, even actually in Indonesia, dalam bahasa Indonesia juga bisa.
Silakan Dek, kalau mau presentasi. Kalau mau tanya dalam bahasa Indonesia. Sulit ya, topiknya agak berat memang ya Pak.
Ini agak berat ya. Oh iya mungkin ya.
Protein ya, aras protein memang mungkin juga adik-adik juga belum begitu memahami juga. Ya sekilas mungkin, ya sekilas mungkin tahu, tapi tidak dalam sekali.
Iya sih betul. Karena sebetulnya untuk untuk memahami ini semua sih butuh apa ya? Kalau ya biochemistry basic mungkin ya.
Karena Pak Kenny background-nya kan dari kimia ya. I think you have a how to say background-nya tepat gitu loh Pak. Kimia, jadi untuk mempelajari tentang struktur protein, kristalografi itu mungkin lebih mudah ya Pak ya.
Iya,
karena
betul.
Memang dari S1, S2-nya kimia ya.
Betul. Iya, saya sebetulnya waktu pertama itu saya tertarik dengan struktur protein itu sejak S1 sih. Ketika dosen saya memperlihatkan struktur protein, langsung wow, keren banget kayak gitu. Kita dengan kita tahu structure, kita bisa memanipulasi protein, melakukan sesuai dengan apa yang kita inginkan atau segala macam, mengoptimasinya segala macam, dan aplikasinya itu bisa kemana-mana kan, ke industri bisa, ke drug discovery bisa, medicine dan lain-lain seperti itu. Jadi maka dari itu saya tertarik sebetulnya.
Ya, Pak, saya ada satu pertanyaan. Kalau tadi kan Pak Kenny menjelaskan tentang S proteinnya, spike protein itu kan related sama binding di ACE ya, ACE2 ya.
Itu ya.
Ya, itu berarti kalau virulensi dari COVID itu, varian COVID itu ditentukan dari ini, S protein atau bagaimana?
Ada beberapa faktor sih. Iya, ada beberapa faktor. Tapi yang pertama itu ya dari S proteinnya itu, karena semakin kuat dia berinteraksi S protein dengan ACE2, itu kan semakin mudah untuk si virus untuk masuk. Faktor yang pertama. Faktor yang kedua itu adanya tadi yang furin cleavage site. Nah, cleavage site itu dia jadi ketika setelah berinteraksi, jadi si protease itu ada protease yang lain yang dari sel manusia itu memotong itu daerah sana. Jadi antara S1 dan S2, region antara S1 dan S2. Nah, ketika dipotong, maka kan si RBD-nya itu dia tetap berinteraksi dengan ACE2. Oke. Tapi virusnya itu rilis dan dia melepaskan bagian S2-nya itu yang berperan untuk masuk kayak drill ke dalam membran dari host cell-nya kayak gitu. Jadi semakin memudahkan untuk pemasukan virus ke dalam sel.
Oh, terima kasih Pak Kenny. Ada lagi mungkin pertanyaan dari adik-adik? Malu ya? Atau mungkin lesu ini karena puasa Pak Kenny?
Iya mungkin.
Izin bertanya.
Ya.
Iya, Patrick ya, silakan.
Kenapa kalau tadi ada mutasi di RBD, habis itu antibody nggak bisa nempel, tapi dia tetap bisa nempel sama ACE receptor?
Oh iya, karena itu kan kalau misalkan kita lihat sebentar. Kalau kita lihat interaksinya itu jadi kalau misalkan di sini ya, contohnya ini ya. Nah, si antibody itu dia kan kompetitif ya interaksinya dengan ACE reseptor kayak gitu loh. Jadi duluan kayak gitu. Nah, ini interaksi ini ditentukan oleh jenis asam aminonya di permukaannya ini. Nah, tadi kita lihat kalau misalkan interaksi antara ACE reseptor dengan RBD ini kan sangat kuat, sekitar 5 nanomolar ya. Nah, biasanya interaksi antara antibody dengan si ini RBD itu enggak terlalu kuat biasanya seperti itu. Jadi kuat-kuatan aja. Nah, apalagi kalau misalkan ada ini misalkan ini mutasi E484K ini itu untuk si antibody interaksi ini sangat penting. Keberadaan E484 ini sangat penting untuk kuatnya ikatan. Nah, tapi ketika terjadi mutasi, terjadi perubahan muatan kan, dari tadinya negatif menjadi positif. Nah, malah jadi saling bertolak belakang, kayak gitu loh. Kan ini berarti kalau di sini berarti ada kemungkinan yang di antibodinya itu berpasangan dengan yang positif, kayak gitu. Ya enggak? Nah, kalau diganti jadi K, jadi kan positif dengan positif, makanya jadinya saling repulsion, kayak gitu, bertolak satu sama lain, seperti itu. Makanya dia lebih resisten kayak gitu. Jadi kalau misalkan dia nggak berinteraksi lagi dengan si antibody, maka dengan ACI dia masih tetap bisa berinteraksi karena region untuk interaksinya itu lebih besar, seperti itu. Cukup jelas ya?
Bagaimana Dek Patrick?
Jelas sih, jadi intinya regionnya yang interaksi sama yang mutasi itu ada region yang interaksi sama antibody, tapi bukan region yang interaksi sama ACE-nya ya?
Kalau yang ini deh kayaknya sama, tapi kalaupun terjadi mutasi, interaksi dengan ACE-nya itu masih tetap kuat kayak gitu dibandingkan dengan antibody, karena mutasi ini sangat penting bagi interaksi dengan antibody. Jadi kalau termutasi jadinya malah nggak bisa berinteraksi lagi kayak gitu, atau interaksinya itu berkurang seperti itu.
Ya jelas.
Kenny. Halo.
Ya, gimana?
Sorry, saya mau tanya ini terkait ini ya. Jadi maybe it's beyond your expertise. Tapi kira-kira ini yang driver utama mutasi itu apa ya? Apakah intervensi? Ya, intervensi itu artinya karena kita mengembangkan vaksin itu, the virus actually responds to that. And then they made mutations to make themselves better at interacting with our ACE receptors gitu. Atau saya nggak tahu ini, saya nggak ngikuti penelitian di bidang ini. Kayaknya ngikuti nggak ini? Drivernya itu apa yang utama gitu loh?
Sebetulnya banyak orang yang berspekulasi yang tadi itu. Jadi karena ada stres kan, sebagai stress response, makanya terjadi mutasi kayak gitu loh, karena ada antibody, kemudian banyak terekspos dengan antibody di manusia, maka si virusnya sendiri berusaha meng-evade imun sistem, ya kan. Nah, tapi hal yang lainnya juga karena ini untuk kemungkinan ya, karena si genom RNA virus itu kan dia nggak terlalu, maksudnya nggak terlalu, the fidelity-nya itu nggak terlalu bagus kayak gitu untuk mereplikasi. Makanya ada kemungkinan terjadi mutasi kan, terjadi mutasinya itu menjadi lebih mudah. Mungkin sebetulnya ada banyak terjadi mutasi, cuman mereka nggak survive aja kayak gitu. Nah, ketika mutasi di bagian ini menyebabkan si virus menjadi lebih survive, makanya dia bisa bertahan dan lebih virulen kayak gitu sih. Mungkin lebih ke arah apa ya, kayak random kayak gitu.
Atau memang mekanismenya gitu ya, mekanisme. Jadi dia tanpa ada represi pun apa namanya, dia behavior memang gitu kayaknya. Virus itu behaviornya.
Iya, virus secara umum kayak gitu.
They mutate themselves periodically.
Iya, seperti itu. Tapi kadang-kadang ada suatu saat itu dia mutasinya itu banyak kayak gitu loh. Jadi makanya kayak jumping kayak gitu.
Iya.
Seperti itu. Bahkan untuk yang UK varian juga dibilangnya itu seperti itu, ada kemungkinan. Jadi ada beberapa mutasi yang tiba-tiba banyak kayak gitu. Mungkin nggak tiba-tiba sebetulnya, cuman karena kita aja nggak tahu kayak gitu loh mutasinya itu di daerah mana, seperti itu.
Dan ini ketahuannya memang di UK ya, populasi UK ya, di tempat lain belum ada belum terdeteksi ada mutan ini, atau memang
sudah di Indonesia juga udah ada sekarang.
Indonesia sudah ada, Pak. Kebetulan ada satu mahasiswanya Pak Aji presentasi itu tentang ini SARS-CoV-2 ini.
Oh gitu. Oh, iya.
Iya.
Indonesia sudah ada.
Iya, sudah mulai ada.
Dan katanya ini lebih virulen dan lebih cepat menyebar. Well, dari segi ininya sih katanya nggak tahu ya. Ada yang bilang dari segi severity-nya itu katanya nggak terlalu severe ke manusianya. Tapi ada juga yang bilang, yang nggak tahu sih sekarang perkembangannya seperti apa. Kalau dulu katanya nggak terlalu cepat menyebar, tapi nggak terlalu ini severe.
Matikan gitu ya.
Cuman nggak tahu juga. Tapi yang ini yang South Africa kayaknya yang lebih parah. Dan sekarang itu kan lebih banyak lagi sebetulnya variannya kan dibanding yang dua ini sebetulnya.
Ya, terima kasih Pak Kenny atas diskusinya tadi, mulai dari struktur perhatian. Terima kasih Pak Tunjung atas pertanyaannya. Selama 2 jam tadi Pak Kenny sudah menyampaikan banyak hal ya, mulai dari yang dasar sekali, yang struktur protein, mulai dari asam amino, dari struktur primer, sekunder, tersier, hingga kuartener, hingga sampai aplikasinya ya, untuk mempelajari atau mengembangkan new drug tadi ya, berbasis protein. Sebelum kita tutup mungkin bisa sesi foto dulu ya Pak Ken ya, untuk kenang-kenangan.
Oh ya, boleh.
Semua mahasiswa, please turn on your camera. We will have a photo session.
Share screen-nya dimatikan dulu aja.
Ya Pak,
saya
silakan
sudah.
Pak.
Sebentar.
Share screen-nya dimatikan dulu Pak Ken.
Oke, sebentar. Udah saya matikan ya share screen-nya. Oh ya, saya belum matikan. Oke. Nah, sekarang saya sudah.
Karena ada banyak ini Pak ya, ada 6, ini saya ambil 6 kali ya.
Oke.
Mohon dinyalakan semua, ada 1. Yang belum pakai jilbab silakan pakai jilbab dulu. 1, 2, 3. Yang kedua ya. 1, 2. Yang ketiga ya. 1, 2. Yang keempat. Yang kelima. Yang terakhir. Oke, terima kasih Pak Kenny atas presentasinya. Terima kasih sekali. We are lucky to have you today.
Thank you. Terima kasih juga atas kesempatannya yang diberikan kepada saya untuk bisa presentasi di sini ya. Mungkin terlalu padat ya topiknya, karena apa ya? Saya kemarin juga gimana ya supaya bisa.
Bukan padat Pak, mungkin sulit dipahami mungkin. Beberapa mungkin sulit. Tapi bagus Pak.
Tapi setidaknya saya ini sih idenya itu ingin memberi overview tentang structural protein dari yang paling simple dasarnya sampai aplikasi yang current saat ini seperti itu. Jadi memang mungkin ya 2 jam itu emang padat sekali sih. Cuman kalau misalkan ada adik-adik atau teman-teman yang tertarik mempelajari lebih lanjut, bisa juga komunikasi dengan saya kalau misalkan mau pribadi. Student juga nggak apa-apa, kontak saya saja, nanti kita bisa belajar lebih banyak kayak gitu.
Ya, ini kebetulan ada yang join staff kita Pak yang sedang kuliah di Austria ya.
Oh,
Jadi ada
dia.
salah satu teman kita Budita.
ya,
Budita,
Budita itu mana Budita? Saya kira itu
Budita
siapa?
join lab.
Saya
Dari
lagi
Vienna
lab
itu.
ini, Pak.
Ya, dan risetnya
Di
mungkin
Vienna.
ada sedikit yang mungkin mirip dengan apa ya, tentang kristalografi. Mungkin bisa nanti share atau diskusi dengan Pak Kenny ya.
Boleh.
Ya. Buditanya
Oh, Budi.
menghilang.
Iya, saya
Udah lama di sana.
maaf saya sedang
Halo.
kerja.
Nah, ini
Di lab.
Masuk tahun ketiga
Oh, masuk tahun ketiga. Oh, di universitas mana?
di
Vienna?
Boku, Pak.
Oh, di Boku. Oh, oke.
Iya, pasti tahu kan.
Ke KBRI ya?
Iya, dikit. Ya gitulah.
Iya.
Pak, nanti kapan-kapan bisa kontak-kontakan.
Boleh. Nanti kita bisa kontak-kontak kalau untuk diskusi lebih lanjut.
Oke. Terima kasih Pak Kenny sekali
Thank
lagi.
you, Kenny. Ya,
Ya,
makasih,
sama-sama.
Kenny, ya.
Nanti ini saya bisa share juga slide-nya. Ini slide-nya lumayan besar, jadi mungkin nanti saya kasih link-nya aja.
Ya.
Drive gitu
Di
ya.
Google Drive ya Pak ya. Ya. Terima kasih Pak Kenny.
Siap, ya.
Sama-sama.
Jumpa.
Terima kasih.
Kalau kesini Finlandia, sampai jumpa Pak.
Iya, nanti kalau ke sini kontak-kontak, insya Allah nanti saya sambut.
Oke.
Oke, saya akhiri aja. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Sampai jumpa.
Terima kasih.
Waalaikumsalam warahmatullahi wabarakatuh. Sampai jumpa.
Saya izin ya, Liv ya. Makasih.
Terima kasih, Pak.
Terima kasih, Pak.
Makasih, Pak.
Pak, saya live ya, Pak ya.
Ya, terima kasih Bu Fre.
Ya. Makasih
Ini kalau
Pak Olive. Ya.
kalau sudah selesai
Ya.
stop record ini sudah ditinggal atau harus kita tunggu?
Oh, itu nanti dia muter sendiri tapi laptopnya jangan dimatikan, Pak. Nanti kalau laptopnya
gitu.
dimatikan lah nggak berproses. Iya.
Jadi ini Zoom saya nggak boleh saya matikan ya?
Oh, enggak, Zoom-nya bisa dimatikan.
Oh,
Zoom-nya
tapi
dimatikan nggak apa-apa, nanti
nyala.
converting-nya tetap berjalan. Ha-ha.
Oke, saya
Ya.
bisa end ya. Oke, terima kasih.
Ya, siap. Makasih semua.